So remember the basic structure of our nephron. We start with the renal corpuscle, where the filtrate is formed, then go through the proximal tubule, down into the descending loop, and then the ascending loop, the distal tubule, and then all of our nephrons are attached to a collecting duct, which goes down in the renal pyramid to the minor calyces. Forming the renal corpuscle is our glomerular capsule that's made up of our parietal layer, which are squamous epithelial cells, and then the visceral layer, which is made up of our podocytes. Remember the filtration slits and the endothelial cells, or the capillaries, are what form our filtration membrane in the urinary system. And here is just the model that we have of the renal corpuscle in lab. Cells along the way, as we saw with the podocytes and the parietal, what the shape of those cells were for filtration, forming their filtration membrane. With the proximal tubule, as you'll see, about 90% is reabsorbed in the proximal tubule of the filtrate. So we have lots of adaptions, such as microvilli on each side to increase surface area to assist in that reabsorption that happens in the proximal convoluted tubule. Then with the thin descending limb of nephron, we have simple squamous cells with some macrovilli. This is where most of your remaining water reabsorption will happen. Then in our thick ascending limb of nephron, we have not as many microvilli. This is where we're going to have the remainder of our sodium and chloride reabsorbed. And then up here in the distal tubule, we'll have secretion and reabsorption. And so we have the modifications not as numerous as with the proximal, where we finish out our reabsorption. And this is where we're going to have our obligatory, or this is obligatory over on the proximal, and this is where we're going to have our facilitated reabsorption, because it is going to be facilitated by those hormones that we've talked about in the past, the antidiuretic hormone and the aldosterone. Another important structure is the juxtaglomerular apparatus, which is made up right here where the afferent and efferent arterioles come together, and that the distal convoluted tubule comes between them. In the distal convoluted tubule, we have the macula densa, and lining the arterioles, we have the juxtaglomerular cells. And in between those two, we have the muscular cells. And these are what are going to start that RAS pathway, the renin-angiotensin-aldosterone pathway. The collecting ducts, which is where the nephrons hook into, also are going to have the ability to reabsorb and secrete. And so they have some of the microvilli to increase surface area and are also influenced by the hormones. And so all of these collecting ducts, as I mentioned before, travel towards the tip of the renal pyramid and enter into the minor calyces. We have two types of nephrons. You have the cortical nephron and our juxtaglomerular nephron. The cortical nephron just barely dips down into the cortical area or the medullary area of the renal pyramid, whereas the juxtaglomerular dips way down into the medullary area. And so this darker area is the medulla, the upper area is the cortex. The capillary bed surrounding the juxtaglomerular nephrons is called the vasa recta, and this is where we get that concentrated urine formed, is with the juxtaglomerular. All right. So there are three main processes that happen during urinary formation. We have filtration formation, reabsorption, indicated by the green arrows with the R, and then secretion, which is delineated by the arrow with the S. So reabsorption is where you take the filtrate and return it to the circulation. Secretion is where you pull things out of the circulation into the filtrate. So let's do a close-up look at that filtration membrane that we talked about before. So here are the foot processes of our podocyte called pedicels. And there is our single endothelial layer and the capillary. And so these are fenestrated capillaries, remember? And so we have these larger areas called fenestrations where larger molecules such as albumin can get through. And so this, with the basement membrane, the basal lamina, forms our filtration membrane. Larger things such as erythrocytes and leukocytes and platelets cannot leave the glomerular capillary and enter into the filtrate. But larger molecules such as albumin and other proteins and water and small solutes can all enter into the filtrate. Common nitrogenous waste that you're going to see are urea, ammonia ions, creatine, produced by the enzyme creatine kinase in the muscles. Remember, that's one of the ways in which we can make ATP. And uric acid, which is a product of nucleic acid metabolism. So these are the nitrogenous waste that are exiting the body via urine. Also, things that will enter the urine: glucose, amino acids, and very small proteins. All right, so let's watch this video on glomerular filtration.
Each kidney contains over 1 million microscopic structures called nephrons. Nephrons are the functional units of the kidney, each one capable of filtering blood and producing urine. Urine production begins with filtration. This process takes place across the filtration membrane, a porous structure located inside the renal corpuscle of the nephron. Filtration produces a fluid called filtrate, which is similar to blood plasma without the proteins. The filtrate collects inside the capsular space of the renal corpuscle and from there enters the renal tubule to be further modified until it eventually becomes urine. Filtration happens at the glomerulus, a ball of fenestrated capillaries located inside a hollow chamber called the glomerular capsule. A layer of specialized epithelial cells, known as podocytes, wrap their foot processes around the glomerular capillaries. Between the foot processes are narrow gaps called filtration slits. If we examine a cross-section of a glomerular capillary, we can also see that between the podocytes and the endothelial capillary wall, there is a layer of extracellular collagen meshwork called the basal lamina. Together, the fenestrated capillary wall, the basal lamina, and the podocyte filtration slits form the filtration membrane. These components of the filtration membrane act as a series of finer and finer sieves, which allow only relatively small molecules to pass through. As a result, filtration is selective based on molecule size. Water and small solutes, such as electrolytes, nutrients such as glucose, or wastes such as urea, leave the blood and collect inside the capsular space to form filtrate.
All right. So here is about the average of our fraction. So about 20%, a fifth of the plasma enters the glomerulus, becomes filtrate in the capsular space. Those glomerular capillaries increase their surface area, allowing for a large filtration fraction. And you filter about 125 mils per minute, or equivalent to filtering all 3 liters of blood about 60 times per day. So you filter your entire blood volume about 60 times per day. And if you recall back when we talked about capillaries with exam 2, the things that affect filtration pressure are the same things that affected the ability of the capillaries to push pressure out in the capillary beds, and that's our hydrostatic pressure and our colloid osmotic pressure. And so net filtration pressure calculated by comparing those forces that are pushing out with those forces that are pushing in. Hydrostatic pressure is going to push out. Our glomerular hydrostatic pressure, which is about 50 millimeters of mercury, will push out. Our glomerular colloid osmotic pressure will pull back in. So that is the solutes left in, such as albumin in the blood, that will pull the fluid back in. And then as you push fluid out into the capillary area, you develop pressure out in this capillary area, so you get capsular hydrostatic pressure that also pushes back in. And so it'll be 50 minus 30 minus 10, which will give you a net filtration of 10. And you can see that here. And so we favor the formation of filtrate because we have a net filtration pressure of 10 millimeters of mercury. And here you can see another depiction of that with the glomerular hydrostatic pressure favoring filtration, glomerular colloid osmotic pressure opposing filtration, along with the glomerular, the hydrostatic pressure within the glomerulus is also, so that is our capsular hydrostatic pressure pushing back in as the filtrate forms in that glomerular capsule. So that means our net filtration is 10 millimeters of mercury.
That filtration moves water and small solutes out of the glomerular capillaries to form filtrate. Now let's focus on the 3 individual forces that determine whether filtration will take place. The first force driving filtration is a strong pressure called the glomerular hydrostatic pressure, or GHP. GHP is the force with which blood pushes on the walls of glomerular capillaries. Recall that glomerular capillaries are fenestrated and therefore leaky. So as blood flows through them, the GHP will drive water and small solutes out into the capsular space. It works much like a garden hose with small holes. As water flows through the leaky hose, some of it will flow out. The higher the water pressure, the more water will shoot out. The GHP is largely determined by the systemic blood pressure, and at about 50 millimeters of mercury is stronger in the glomerulus than in any other systemic capillary. 2 forces oppose filtration. One is capsular hydrostatic pressure, or CHP, and the other is glomerular colloid osmotic pressure, or GCOP. The CHP is the hydrostatic pressure of the filtrate as it accumulates inside the capsular space and pushes on its walls. This pressure of about 10 millimeters of mercury tries to push water back into the glomerular capillaries and therefore opposes filtration. The second opposing force, GCOP, is due to the osmotic gradient created by large solutes that remain in blood. This pressure pulls fluid back into the capillaries by osmosis. It is a force of about 30 millimeters of mercury and opposes filtration. Think of the 3 forces as playing tug-of-war. As long as the GHP is stronger than the combination of CHP and GCOP, there will be enough pressure to drive filtration. We combine the 3 forces to yield the net filtration pressure, or NFP. The NFP drives filtration. We can measure filtration by using the glomerular filtration rate, or the GFR, defined as the amount of filtrate produced by both kidneys per minute. The GFR indicates whether the kidneys can successfully clear toxic wastes from the blood. There are many homeostatic mechanisms that control the GFR and ensure proper filtration.
All right. So before moving on to the factors that actually affect glomerular filtration rate, we'll stop this video and continue in a second video. Let me know if you have any questions.